So today we're going to talk about HIV and AIDS. So not the most enjoyable lecture right after a holiday, break, but HIV, as you know, stands for human immunodeficiency virus. AIDS is the manifestation of that disease, acquired immune deficiency syndrome. In 1981, in the United States, there was a cluster of unusual infections and diseases. So pneumocystis pneumonia is a fairly rare type of pneumonia, and normally it's not seen in healthy people. Same thing with Kaposi sarcoma. And remember, we talked about Kaposi sarcoma. It's caused by human herpes virus number 8. And it's one of those viruses that many people are infected with because it's transmitted through saliva, but they normally do not show any symptoms or present with any problems as long as they have a healthy immune system. But in a group of young homosexual men, these two conditions were seen, and they never occur in young people. And so the question is, why are they occurring in these young men, and in particular these young gay men? And so what they discovered about these men was that they had a loss of immune function. So their immune systems were not functioning properly, which is why they had become susceptible to these conditions. And so the scientists realized there must be something causing this. And in 1983, HIV was officially discovered by Luc Montagnier at the Pasteur Institute in Paris, France. Although the United States were close behind them, and they were also working on the discovery of this virus, the French get the official credit, and HIV was the official name that was settled on. Initially, there were a couple other names for it, but they finally settled on HIV. This was in 1983. So the origin of AIDS is believed to have crossed the species barrier into humans somewhere in Africa in the 1930s. The earliest known patient who died of HIV was in 1959 in the Congo. And so the question is, how did they know about a case in 1959 when they just discovered it in the early 1980s? So any idea as to how they might have, figured this out? A patient from 1959 when they only discovered HIV and AIDS in the 1980s? Anybody have a guess? Any ideas? How they could have known from 1959, right, that a patient died of AIDS? No ideas? Well, one of the things that doctors and scientists often do is save blood and tissue samples. And so what happened is the scientists started going back and testing all those blood and tissue samples looking for HIV, and the oldest known positive case came from 1959. So HIV had been around for a long time. It's just that not that many people were dying from it. And of course, when you die from AIDS, what you die from is some infection that you cannot control. And so these deaths did not look at all unusual, right? Someone died of pneumonia, right? Someone died of this infection, someone died of that infection, but it was nothing out of the ordinary. It wasn't until we had clusters of cases of unusual infections that scientists took notice that something was going on. But it's believed to have spread throughout Africa as a result of urbanization. As more and more people from rural areas moved into the big cities, HIV was spread more easily that way. because whenever you have high population densities, all kinds of diseases spread more easily and at higher rates. And it's believed to have spread throughout the world as a result of modern transportation, so people taking planes and boats and then engaging in unsafe sexual practices. Now, in 1976, there was a Norwegian sailor who died of HIV and AIDS, and he's believed to be the first known case in the Western world. And actually, he was erroneously labeled as patient 0. And patient 0 refers to the index case, the first case that's believed to have caused the spread of an infection. And it was actually sort of a clerical error where his numerical designation simply started with 0, but then people started abbreviating and saying patient 0, and then he got falsely labeled as patient 0 as the one who brought HIV to the Western Hemisphere. And that was not true. Later, long after he died, he was exonerated of being patient 0. And there were many people who brought HIV to the Western Hemisphere. So the origin of HIV is controversial because it's believed to have originated in Africa. Many Africans don't like that distinction, but it is a newly emerged disease, so it probably hasn't been around as HIV much longer than 50 to 100 years as HIV. And there have been many possible origins suggested for HIV, but 3 of the most common. 1, it's a man-made biological warfare creation, and we'll look at evidence for that later. Number 2, it's a mutated monkey virus that crossed over into humans, and this is the most widely believed hypothesis as to the origin of HIV. Or 3, maybe HIV has been around for a very long time. It's just spread very slowly. It killed very few people, so it was never noticed until recently. But there is a monkey virus, SIV, the simian immunodeficiency viruses, which is very genetically similar to HIV. So it's believed that HIV mutated from SIV. And it's happened within the past century. And of course, today, HIV infection is pandemic. And what is a pandemic?
Worldwide?
Yep, a worldwide epidemic. So if you did not know that word before, you know it now with coronavirus, right? So some characteristics of HIV, it is surrounded by a lipid membrane. Where did that lipid membrane come from?
The host cell.
Right, the host cell. So HIV steals membrane from the host cell. And it does have protein spikes. A GP stands for glycoprotein spikes. And it uses these spikes for attachment, right, to the host cell. It has an RNA genome, and there are 2 copies of the RNA genome consisting of at least 9 genes. So it's 9 or 10 genes. So when we look at everything that HIV does, it does so with only 9 or 10 genes, right? Compared to humans that have at least 30,000 genes, right? So viruses have a small number of genes, and yet they're remarkable in terms of what they can do. The virus also carries 3 proteins with it. It brings the enzyme reverse transcriptase because HIV genome is RNA, so it needs to make a DNA copy. So it brings a reverse transcriptase with it. HIV also brings the enzyme integrase with it because now that HIV has made a DNA copy, it needs to insert that DNA into a host chromosome. So the enzyme integrase does that. And then protease is a specific enzyme that HIV needs to cleave some of its viral proteins. But HIV must bring these enzymes with it because host cells don't have these, or not the specific kind of enzyme that HIV needs. So in addition to its RNA genome, HIV carries 3 important enzymes with it. All 3 of those enzymes are targets for antiviral drugs. And as I already mentioned, HIV is very genetically similar to SIV. So here we see our lipid membrane envelope and the glycoprotein spikes. And they're actually made of two proteins, the larger protein and a smaller protein. The larger protein has a molecular weight of 120 and the smaller one of 40. So that's how you get 160 as the total weight for the attachment protein. And HIV's capsid is unusual in that it's cone shaped, so it's a conical capsid. So it's not a typical type of shape for a viral capsid. And we notice it's 2 copies of the RNA genome, and most diagrams only show reverse transcriptase, but the other 2 enzymes are also there with it. So of course, the first thing that any virus must do is attach to its host cell. And the host cell has receptors on it that HIV uses, right, to attach and gain entry into the host. You do not need to memorize these receptors, but the main idea here is that 2 cell surface receptors are required for HIV to attach and get engulfed by the host cell. So 2 cell surface receptors, that's very important. And these cell surface receptors are found primarily on immune cells like T cells. They can be found on certain other cells in the body, but it's mostly your immune cells that are the targets for HIV. Once HIV attaches, the host cells will engulf them, right? Receptor-mediated endocytosis. The capsid breaks apart. The RNA genome is released. Reverse transcriptase makes a DNA copy. Then the integrase enzyme integrates that HIV DNA into a host chromosome, forming a provirus. So remember, in bacteriophage infections, we call the integrated virus a prophage. Here, we call it a provirus. And the other difference here is that the provirus never leaves. It is a permanent part of that host chromosome. So every time the host cell divides, each daughter cell has a copy of that provirus permanently, right? It never leaves. And since it's now DNA, the DNA is transcribed and translated, right, to make virus proteins. And when the HIV is transcribed into RNA, that is the genome, right, for HIV. So that's how it makes more copies of itself. And then the viruses assemble and bud from the host, stealing host membrane, eventually killing that host cell. Initially, early in the infection, lots of viruses bud like crazy, so they kill, T cells especially rapidly in the very beginning. But then the rate of new HIV virus particle production decreases, and it happens more slowly. And so cells can survive a lot longer and constantly produce new HIV particles. So as I mentioned, the host cells are infected for life. So as long as that host cell survives, it's infected and producing new HIV particles. But HIV doesn't have to be floating around through the bloodstream, where it can be attacked, right, by the immune system or by antibodies. It can be hiding in lymph nodes. Remember, lymph nodes, you have congregations of T cells and B cells in your lymph nodes. And so it's very easily, right, to spread HIV from one cell to the other in a lymph node. And macrophages are also infected by HIV. And remember, what's the job of the macrophage? It's supposed to find things, engulf them, digest them, and present antigen to a helper T cell. Well, HIV is one of those pathogens that avoids destruction inside of a macrophage. So when macrophages engulf them, HIV does not get destroyed, but the macrophage goes to a helper T cell and says, look what I found. So the macrophage delivers HIV to helper T cells to infect them. So Basically, the immune system is infecting itself with HIV while trying to do its normal job. So here we see HIV, and again, notice 2 cell surface receptors are required for attachment. So you don't need to know specifically what those receptors are, but the key point here is that a cell must have these 2 receptors on the cell surface to get infected with HIV. So once HIV attaches and penetrates, the RNA genome gets released, it's reverse transcribed into DNA, it's integrated into a host chromosome, forming a provirus. When the provirus is transcribed into RNA, that's the genome. When the RNAs get translated, those are the viral proteins. Everything assembles, and the virus is bud from the cell-stealing host cell membrane. As I mentioned early in the initial infection, the rate of cell death is pretty high because the viruses are budding like crazy. But over time, the rate of new viruses produced does slow down. And so those infected cells survive a lot longer and are constantly producing new HIV particles. Yeah, don't worry about that one. So monocytes and macrophages serve as HIV reservoirs. So once these cells get infected with the entire virus, they carry that virus with them wherever they go. Eventually, the helper T cells, which are the main cell type that get infected and destroyed, they decline, and you get immunosuppression, right? Your immune system has become crippled. And when it comes to the number of T cells per cubic millimeter of blood, different sources cite different numbers, which is why you see a broad range here. But in general, if your T cell count drops below 200 cells per cubic millimeter of blood, that is one of the criteria for HIV infection that has become AIDS. So at that point, your immune system cannot function well enough. When it comes to a normal range of T cells, again, it's a broad range because different sources cite different numbers, but at least 500 to 1500 cubic cell or cells per cubic milliliter of blood, and that's a normal T cell count. So if you're below 500, you'll start becoming more susceptible to infections, and once you reach below 200, you meet the clinical definition of AIDS, and at that point, you're not able to fight off, most anything, so you succumb to some kind of infection. Even cancers like Kaposi sarcoma, right? Because helper T cells help to stimulate cytotoxic T cells, which can kill cancer cells. So remember the central role of the helper T cell, how it stimulates B cells and cytotoxic T cells. And when you eliminate helper T cells, you become immunocompromised, right? Your immune system is crippled. But also remember, it's possible for B cells to interact directly with antigen and then proliferate and differentiate and make antibodies. But that's not as efficient a process. But if you have memory B cells, to a past pathogen, you should still be able to resist that pathogen. Even if you do have AIDS, it's just new pathogens you may not be able to resist. So we talked a lot about antibodies, and antibodies do get produced against HIV, although in some people, and I'm not quite sure why, but they may be slow to produce the antibodies, which is why, you need to be tested at least 3 months after you may have been exposed, because in some people, it takes 3 months to make detectable antibodies. So even though you should be making them in 7 to 10 days, in some people, the antibody response was slower. But antibodies do get made. So in people infected with HIV, they are making antibodies, but the antibodies don't really help. And the question is, why not, right? We've just seen how wonderful antibodies are. Well, once HIV has infected a cell and formed a provirus, antibodies cannot do anything against the provirus, right? So those cells are still infected and still making new HIV particles. We just saw that HIV doesn't have to be floating around through the bloodstream. It can be hiding in lymph nodes. It can be hiding in macrophages, again, where antibodies can't get to it. But one of the big problems is HIV's mutation rate. Reverse transcriptase is nowhere near as good as DNA polymerase. So as HIV reproduces, many mutations occur. And those mutations keep changing the antigens on the surface of HIV. And by some estimates, and these are just numbers kind of to illustrate the potential problem, is that assuming that someone receives no HIV medication and they survive up to 10 years, it's possible for that person to have produced as many as 10 trillion virions, so 10 trillion new viruses during their lifetime. When you're talking about something that only has 9 or 10 genes, what that means is that every possible mutation that can occur will occur. And it's simply not possible for the immune system to keep up with that rate of mutation. Because even if you're making new antibodies in 7 to 10 days, in that period of time, HIV has mutated just enough. There's at least a few particles which are different enough that those antibodies are no good. And so you have to make more antibodies. And that takes another 7 to 10 days. And by then, HIV has reproduced and mutated again. So there's some new particles that have changed just enough that those new antibodies don't work. And so on and on and on. And it's simply impossible for the immune system to keep up with that rapid rate of mutation. That's also why a single drug, monodrug therapy, is never going to work, because chances are there's already going to be at least a few HIV particles resistant to that one drug. And that's why a combination of drugs are being used, not to cure, but to suppress HIV replication. And that's also why we still have no vaccine. We've known about HIV since the 1980s. It's been thoroughly sequenced and studied. Still, there is not an effective vaccine against it. And that's, surprising, especially in less than a year, we've created a vaccine against COVID-19, but here we've had almost 60 years and still no HIV vaccine that works. So HIV is like nothing we had ever seen before. So when it comes to the stages of HIV infection, category A is where you're basically asymptomatic. Upon the initial infection, you get flu-like symptoms. So that's, very common. We've talked about lots of viruses, the initial infection, you get flu-like symptoms. So it's nondescript, right? Non-specific. And you may have some persistent swollen lymph nodes, but if you suffer from allergies, that's not uncommon either. When you progress to category B, you get things like persistent Candida albicans infections. These are yeast infections. In the oral cavity, it's called thrush. And once you progress to category C, you've now met the clinical definition of AIDS. And you start becoming susceptible to all kinds of things like pneumocystis and Kaposi sarcoma, all of these dormant herpes viruses, right, that you're probably infected with, like cytomegalovirus, re-emerge and can be extremely nasty. If you have dormant TB, and next lecture we'll talk TB, if your TB is dormant, it can be reactivated because now your immune system is crippled. It's not able to keep TB suppressed. And toxoplasmosis, a protozoan infection that often comes from cats, is something that can also happen to people. And you should look up toxo because it's pretty interesting. The crazy cat lady syndrome is a real thing because they're infected with toxoplasmosis and it affects the brain and it makes you kind of schizophrenic. So it's a real thing that can happen. So this diagram is kind of complicated. So let's just focus on the red line and the black line. And so the black line indicates the amount of HIV in the blood. And so upon the initial infection, your blood will be teeming with HIV particles because HIV is just reproducing like crazy. And since it's reproducing like crazy, it's killing your helper T cells in massive numbers. So you get a considerable drop in your T cell count. But the immune system does rebound. It is trying to fight. Your T cell count goes back up as the amount of HIV in the blood goes way down. It never disappears. It's there at a low but constant level. And so if you look at the number of years, and for most people, before we had any anti-HIV drugs, they would survive for 5 to 10 years right before finally dying of AIDS. And so during that 5 to 10 year period, they are constantly producing HIV, right? So they're infectious, and their T cell count is slowly declining. But it's still enough T cells to maintain a healthy immune function. And so all during this period of time, these patients are asymptomatic, right? They have no symptoms, no idea that they're infected with HIV. And so they appear healthy. And so because they appear healthy, what are they doing during these years? They're spreading HIV because they have no symptoms, right? So they don't know they're infected. And this is one of the reasons why HIV spreads so quickly, is because many people were infected for years before they showed symptoms. And during that period of time, they thought they were healthy, so they did not take any precautions. And so they were spreading HIV. But at some point, their T cell count drops just below 200, and they meet the clinical definition of AIDS, and they now began to manifest symptoms. But for many years, these people look and feel healthy. And so they're spreading the virus because they don't know, right, that they're sick, right, that they're infected. And that was one of the problems with HIV. It had a fairly long incubation time where you were still healthy, but you were infectious. And it's not typical to be infectious for years without showing symptoms of that infection, but that's one of the things that HIV does. And so that's why it spread, so quickly throughout the world population, right? Because if you think you're healthy, you don't take precautions. And this was just an average, most people would live 5 to 10 years right before they died. But there were cases where, people got infected and they were dead within a few months, right? Because their immune system just could never bring HIV under control. And so they did die very quickly. And then on the other end, you have some people that were called long-term non-progressors. So it may have taken 20 or more years before they finally progressed to AIDS. So HIV infection, has a wide variety of manifestations. Not all human beings are equal. Not all human beings were equally susceptible. Not all human beings progressed at the same rate. And so that made HIV even more difficult, right? Because it wasn't the exact same for everyone. And there was a lot of confusion back in the 1980s, and it was a scary time because they just didn't know enough about HIV yet. And there were conflicting, reports because not everyone was reacting the same way to HIV infection. So it was a scary time because they just didn't know that much about it. Now, when it comes to HIV's survival outside of a cell, it is a very sensitive virus, right? All viruses that have lipid membranes, it's very easy to damage those membranes. And as soon as the membranes are damaged, those viruses are no longer infectious. And so HIV outside of a cell, it's subject to drying very quickly. But that's if it's just, plain HIV. If it's in a bodily fluid, even a dried, bodily fluid like blood or semen, it can last a whole lot longer, maybe up to 6 days. So again, that's kind of unusual. But inside of a cell, once HIV gets engulfed, it's quickly broken down to release the RNA genome. So basically, in less than 2 days, the HIV particles are no longer there, right? The infection is well on its way. Now, HIV is transmitted through bodily fluids. So remember, we talked about HIV as being, it is an infectious disease, but it's not easily, right, transmissible. It's not contagious, right, because you do have to exchange bodily fluids in order to get infected. So just being in a room with someone is not dangerous. Just touching them, ordinary physical contact was not dangerous. It required an exchange of bodily fluids, and most commonly through unprotected sexual intercourse, vaginal, anal, even oral intercourse, especially if you have gum disease or gingivitis, because that created a portal of entry, right, for HIV infection. Breast milk is sort of controversial because at one time it was believed that if the mother was HIV positive, she should not breastfeed her baby. But it turns out that may be safe, at least in certain women, because a milk, as we learned, it does produce antiviral compounds, which may help to neutralize HIV. If a woman is HIV positive when she gets pregnant, then the fetus often gets infected with HIV. But one of the first drugs used to treat HIV was AZT, which, as it turned out, was not really very effective at treating AIDS patients, but it is very effective at preventing the fetus from getting infected with HIV. So if a woman is HIV positive and gets pregnant, she can take AZT, and that will greatly reduce the chances of her fetus getting infected. And AZT has been around a long time now, so it's fairly cheap and affordable, right, for most women all over the world. Blood contaminating needles. Intravenous drug use is the second most common mode of transmission for HIV, the first being unprotected sexual intercourse. But IV drug users have high rates of HIV infection because they share needles, right, to inject their drugs. And if one person is infected with HIV, the needle that was used has blood, right, from that person still in it. And so that gets shared and transmitted to other people. Organ transplants and blood transfusions. In the 1980s, this was a way that HIV was transmitted, but it's not an issue today because we have excellent testing today. So there's no worries about blood transfusions or organ transplants. But back in the 80s, before they could test for HIV, people did get infected right, through blood donations and organ transplants. And there is some controversy because when there was the very first sort of primitive test for HIV that was available, it was not used right away. And that delay allowed many people to get infected with HIV through blood transfusions. In particular, hemophiliacs, because hemophiliacs, they do not have clotting factor, and so they use blood donations, right, to extract the clotting factor, which is what is used by hemophiliacs. And so if only one of the donations was infected with HIV, all the clotting factor made was infected with HIV. And that clotting factor was distributed to hemophiliacs all over the world. So many hemophiliacs got infected with HIV and died of AIDS when their deaths could possibly have been prevented by using that early test for HIV. And that did not happen right away. But it's not a concern today. And then artificial insemination. You know, some women choose to get pregnant by using a sperm donor. Again, if that person was infected with HIV, his semen had HIV in it. And so women got infected that way. But not a common mode of transmission today. As I said, unprotected sexual intercourse and IV drug use are the most common modes of transmission. HIV is a non-discriminatory virus. It doesn't care who you are, your race, your ethnicity, your religion, your economic status. HIV was an equal opportunity pathogen. At least that's what was believed in the 1980s and into the 90s. But it turns out not everyone is equally susceptible to HIV infection. There are, in fact, humans that are resistant to HIV. And these humans are mutants. They have a mutation in one of the cell surface receptors, in particular the CCR5 receptor. So those mutant human beings cannot get infected with HIV because they only have one of the two receptors needed. So they're resistant naturally. And so that was a remarkable finding, right? That some people are actually resistant to HIV because they only have one cell surface receptor. And we've been saying HIV requires two cell surface receptors. If you only have one, then you cannot get infected, right, with HIV. So here's the controversy, and one of the reasons why some people suspected that HIV was a biological weapon. When we look at the distribution of HIV all over the world, we find the majority of cases are in sub-Saharan Africa. So Africans are primarily affected by HIV when we look at the total numbers all over the world. And it turns out that the resistant mutants are white people of European descent. And so some speculated that HIV was a weapon created to wipe out Africans. But the problem with that theory, in addition to just being ludicrous, is that the number of resistant white Europeans is small. And if you remember your genetics from 150, we have two copies, right, of a gene, and we call those copies alleles. And so if you're homozygous for the mutation, you don't produce any of the CCR5 receptors. But if you're heterozygous, you've got one good allele and one mutated allele. And so you do produce some of the receptors. So some of your cells can be infected. So in those people that were heterozygous, they may have required multiple exposures before they actually got infected, or once infected, they were slow to progress. But it's a small percentage. About 1% of people of white European descent are homozygous for this trait, and about 20% are heterozygous. So in order for the theory of a biological weapon to be valid, you would have to believe that white people were perfectly willing to wipe out 99% of white people while trying to wipe out 100% of black people. And that just makes no sense. Now, this mutation was a random chance event, right? A spontaneous mutation, which occurred in the past, and it does provide protection against something else, and I can never remember which one. It was either smallpox or bubonic plague, but those two infections ravaged Europe, and so this random chance mutation helped to protect Europeans from either smallpox or bubonic plague, and that's why that mutation exists. And it's just by coincidence that it protects against HIV. But just a random chance spontaneous mutation, right, which happened to protect Europeans against another infectious disease, it just by coincidence happens to protect them against HIV. And since that discovery, they've looked for other kinds of mutations, and there are some other mutations that exist, which again can reduce the chances of getting infected by HIV, or in some cases, like the Duffy antigen, D-U-F-F-Y, the Duffy antigen, which is found in many Africans. The Duffy antigen evolved to help protect against malaria. Unfortunately, it does make them more susceptible to HIV infection. So again, when it comes to genetics and HIV, things are complicated, right? There are many different mutations, many different variations, which affect how easily someone gets infected or how quickly they can progress to HIV. And there was even a population of prostitutes. I think they were in Kenya, and early on in the pandemic, it was discovered that these prostitutes, which did not use condoms, they were repeatedly being exposed to HIV, but they were seemingly not getting infected. And they thought that maybe these women had some kind of natural protection that we could, learn about and exploit to help protect other people. But it turned out that they did not. And they did not because those prostitutes who also used IV drugs, they got infected just as easily as anybody else. And it turned out the reason why they were not getting infected through sexual intercourse, through vaginal intercourse, is because they actually had a very weak inflammatory response in the vagina. And remember, one of the functions of inflammation is to bring the immune system to that site of infection or injury, right? To bring the macrophages and everything else there. And because they had such a weak inflammatory response, macrophages and dendritic cells were simply not getting infected by HIV. And so it was just much harder for them to be infected through the vaginal route. So again, there's all kinds of unusual variations which affect HIV infection and HIV progression. Of course, HIV is a pandemic. It's all over the world. It's in every continent. As I mentioned, sub-Saharan Africa was particularly hard hit, but when it comes to the numbers of men and women infected with HIV all over the world, and we're looking at worldwide statistics, there are equal numbers of men and women infected with HIV. That makes HIV a heterosexual disease. Not the gay disease that everyone likes to call it, but worldwide, equal numbers of men and women are infected, which makes it a heterosexual disease. It's just that in North America and Europe, it was primarily spread through the homosexual population in gay men. But all over the world, we see equal numbers of men and women infected with HIV, and that makes it a heterosexual disease. We see here for the relative distribution, As I mentioned, sub-Saharan Africa is disproportionately affected by HIV. Part of the problem in South Africa, they had a president, Mbeki, who was an HIV denialist. And so he denied HIV, he denied it was a problem. He told his people not to use Western drugs to treat HIV. And so that led to the massive spread of HIV throughout the African population. We also see high numbers in Southeast Asia, and part of the problem for that, in this country in the 1980s, there was a huge stigma associated with HIV infection because everyone just assumed that you must be gay if you got infected or you were an IV drug user. And so wherever there's still a social stigma against gay people or against drug users, they don't want to let people know that they're infected with HIV. And so they don't even want to go get tested. And so when you try to keep things like that hidden, it just allows it to spread. That's one of the problems in Southeast Asia, especially in China, was the stigma associated with HIV infection and the implications. And so whenever you can't be open about things, they spread. And here's a more recent update. So this is different from your PowerPoint presentation because I made some updates showing the relative spread. And again, Africa is still the hardest hit continent overall when it comes to HIV infection. Again, China, just showing you the relative distribution. As the disease spreads, and certain provinces, like the Yunnan province, have extremely high rates of HIV infection. And again, the problem is the stigma associated with being infected with HIV and what that implies. And so if you're afraid, you don't get tested. If you don't get tested, you spread the infection to others. There are two major types of HIV. The stuff that's in the light gray here, don't worry about that, but there are two major strains of HIV, HIV-1 and HIV-2. HIV-1 is the most common strain all over the world, and it was the first one discovered. HIV-2 was discovered later and is seen primarily in Western Africa. But there are subtypes of HIV. And because all HIV is not the same, that's also one of the reasons it's made vaccine development difficult, right? Because not all HIVs are exactly the same. HIV is entirely preventable. How can we prevent the spread of HIV? What can you do?
Using condoms?
Use condoms, right? And there are condoms for men and condoms for women. So there are female condoms. What else can you do?
Not reusing needles?
Right, if you're an IV drug user, do not share your needles. And there are needle exchange programs where drug users can go and exchange their dirty needles for clean needles. Although there's some controversy associated with that. Certain groups, especially conservative groups, think that's simply encouraging people, right, to use drugs by giving them clean needles. But, those people are going to use those drugs no matter what. So it's better for society if they're not infected with HIV. You can get tested, know your status, right? We have drugs now, which you can take for pre-exposure prophylaxis to help prevent you from getting infected in the first place. Initially, those drugs were used to treat rape victims or healthcare workers who may have been exposed. Now they're more widely available, right, to everyone who may want them. We have lots of very effective HIV medications. In fact, the HIV meds are so effective today that if you've been on them for at least 6 months and the level of HIV in your blood has dropped below the ability to detect it, you are actually considered safe, non-infectious. So you can have unprotected sexual intercourse with someone, and the risk of transmission is extremely low. No one's ever going to say zero, but it is extremely low. And that is a remarkable place to be right now compared to the 1980s. So there are drug regimens where you can suppress HIV replication so well that you're not producing any HIV particles, which means that you cannot transmit HIV, or at least it's very difficult now to transmit it if you're HIV positive, as long as you're on the meds and take them every single day, and you will have to take them for the rest of your life. But we'll get to meds more in a minute. For healthcare workers, since many of you are planning on going into healthcare, you need personal protective equipment, and so PPE. And we know a lot about that now with COVID-19. So gloves, gowns, masks, goggles. If you inject someone, with a vaccine or anything else, you don't put a cap back on the needle. You put the needle into a sharps container. Of course, in very well-developed countries, they have these fancy retractable needles. So after you inject someone, the needle retracts back into the syringe. Those needles are very expensive, right? So much of the developing world cannot afford to use those kinds of syringes. But in general, do not put caps back onto needles. Put them right into a sharps container where you can't touch them. Although the risk of infection from a needle stick is low, there is a risk, and people have gotten infected that way. So when you're dealing with patients, especially any blood from patients, you have to think very carefully. And one of the things that I would reinforce if we were meeting face-to-face is that in lab, I would constantly correct students, for mistakes that they were making. And sometimes students would get offended that I was constantly correcting them. But the reason why is if you're going into healthcare, you can't afford to make mistakes, right? Because you're going to injure or infect yourself or injure or infect a patient. So you have to pay attention to everything you're doing. And as an example, one semester, I had a student, she was my student the previous semester, but she emailed me in a panic because she was working in a clinic and they were doing blood glucose testing, where you prick the finger, right, to get the blood, and then you put a cotton ball on the finger, right, to soak up any blood. And so the patient with whom she was working was HIV positive, and they were doing the blood glucose, and the patient dropped the blood-stained cotton ball, and the student grabbed it with her bare hands. And then she freaked out because she wasn't wearing gloves. And my question to her was, why weren't you wearing gloves? Because you're doing, a blood glucose test with blood. You should be wearing gloves. Now, she was okay, but what she did was something that she should not have done, right? The patient dropped a cotton ball, and she instinctively reached out to grab it, not thinking that it's got blood on it, and she's got bare hands, right? So you do have to pay attention to detail, because even though it doesn't really matter that much in lab, when you leave here, it's going to matter a lot. And so that's why I would constantly correct students for their technique, because it does matter. And unfortunately, you guys are not getting that experience in lab, but if you go on to take nursing courses, you will be meeting face-to-face because you need that hands-on interaction to learn those techniques. But it is important to pay attention to everything you do as you're doing it, because you're putting yourself at risk and potentially patients at risk. So I already mentioned that vaccines, we don't have one yet. A big problem is the high mutation rate. There are different types of HIV. And so if you've been listening to the discussion on the COVID-19 vaccines, the most logical target, right, for a viral vaccine are the attachment proteins, right? Because all viruses must attach. If you can block the attachment proteins with antibodies, then those viruses have been neutralized, right? That's one of our mechanisms, right, of antibody action is neutralization. And so if you cover, right, the viral spikes with antibodies, then you neutralize that virus, right? Yes, right. Sorry, we're going long today. So again, the glycoprotein spikes for HIV were the logical target. And that didn't work. And the reason why, and I don't know if you guys can see me, but if my fist is the glycoprotein spike, and so the top of my knuckles is where the glycoprotein spike is going to attach to those host cell receptors. And so the idea is to cover that spike with antibodies. That would prevent it from attaching. Well, that didn't work. And the reason why, it turns out that the part of the spike that binds to the cell surface receptors is not exposed. It's actually covered by a little protein flap. And so the protein flap can mutate and change, and that's no big deal. But the way it works is, right before the virus goes to attach to cell surface protein, the flap moves out of the way and allows the spike proteins to attach. And so because the spike was protected, there was simply no way to get antibodies there right before the spike bound to the host cell receptors. And so that's why antibodies against the glycoprotein spikes have failed. So HIV is not like anything else that we've ever seen. And so we still have no vaccine. And of course, once the provirus is established, antibodies don't do anything, right? The only way to get rid of the provirus is to destroy those cells. And those cells are your helper T cells. So if you destroy them, you're doing the same thing that HIV does anyway. So once HIV gets established, antibodies are useless. And so that's why, treatment to cure someone has been almost impossible. So, and then almost every year there's some new HIV vaccine in clinical trials that's very promising and ends up being a failure. So, and we do have a huge number of drugs, at least 37 drugs currently available. And you do have to take a combination of these drugs, but the point of these drugs is to block HIV from replicating. So once you're infected forever. And so the goal is stop HIV from replicating, and then your immune system stays healthy. And as I said, we have amazing drugs today which do that, which so completely stop HIV from replicating that you are considered to be non-infectious. And so as long as you stay on those drugs, you will live a normal, healthy lifespan. But that's only because we've done so much research in creating these drugs. But they are remarkably effective today. And although it is a combination drug, it is one pill a day. So one pill a day. So it's like taking a vitamin, a large vitamin, which is kind of a problem because some people think, HIV, it's no big deal. If I get infected, I'll just take this pill every day and I'll be fine. And that's why HIV is still continuing to spread because people are not taking it as seriously as they used to. Not everyone can take these drugs. They do have side effects. So it's not, perfect and wonderful for everyone. But in general, for most people, these drugs are pretty wonderful. Truvada is a prophylactic drug. It's the PrEP You may have seen this advertised, take these drugs for PrEP. And PrEP stands for pre-exposure prophylaxis. So prior to being exposed to HIV, if you're on these drugs, they can prevent you from being infected with HIV. It's not 100%, but it does dramatically decrease your risk of getting infected with HIV. And so again, we have these drugs to help protect people from getting infected, especially important for victims of rape or for healthcare workers who may have been exposed, or they can start taking these drugs immediately, and that can prevent them from getting infected. So there is tremendous hope. No cures, but there are such effective treatments that you're basically non-infectious and will live a normal, healthy lifespan. Now, there has been at least one documented case of someone being cured of HIV. Has anyone heard of that? Someone actually being cured from HIV, Karen?
No, I was just saying I hadn't heard of that before.
Well, so far there's only been one clearly documented cases, possibly two, but this person had something like leukemia, and so they needed a bone marrow transplant. And so it turned out that the bone marrow donor was one of these receptor mutants. And so the bone marrow that was transplanted and then creating a brand new immune system, that immune system was immune to HIV because only one of the cell surface receptors was present. So that patient who was previously infected with HIV has now been cured because of a bone marrow transplant from a donor who had the receptor mutation. And so students always ask, well, then why can't we do that for everyone else? Well, one, bone marrow has to be extremely well typed and matched, and there's simply not enough available donors, right, to match everyone. And two, you do have to then suppress part of your immune system when you receive that bone marrow transplant because you're receiving that foreign immune system into your body. So you must suppress, right, part of that immune response so the bone marrow doesn't attack you. So it was a rare case where someone was actually cured only because they had a terrible disease like cancer that had to be treated with a bone marrow transplant. And so they were cured of their HIV, but it's not a way to cure everyone. It's just not an effective way to do that. But it has happened at least once. But that was the exception. So any questions or comments? I know we went long, but lab this week is kind of short. So any other questions or comments? So you still need to take precautions. You still need to use protection, but we have the potential now to really dramatically reduce the spread of HIV because even people who are infected with HIV, it's so well suppressed that it's hard for them to spread it to other people. So hopefully one day we will eradicate HIV, but we still have a long way to go. But at least people now can survive and live healthy, normal lives. At least for those countries that can afford these drugs. And that's another problem, right? Not everywhere, can they afford all these expensive drugs. So anything else, questions or comments? Okay, so we'll stop here.